All right, guys. So welcome to today's lectures. Today we're going to be talking about randomization and blinding. We've been going through this lecture series. This is actually part of the start of this is part of the clinical. This is actually part of the conduct phase of your lectures, of your practice of clinical research. So randomization is, of course, the learning objective for this class will just be first to understand the concept of randomization. Second, we're going to be discussing the importance of randomization in clinical trials, and also we're looking at the details formats of randomization. There are different kinds of randomizations, right? There are different types and the way they fit into different forms of clinical trials. So number one, we're looking at randomization is the process of assigning clinical trial participants to different treatment groups. Remember, in a couple of lectures that we've had, to actually elaborate it, where randomization, where subjects are being randomized, being placed into different cohorts, or we call them groups or cohorts or whatever. But the most important thing for you to understand is that randomization gives a fair chance for every participant to either be randomized into a fair chance for every participant to participate in the clinical study. And the reason for that is because in the past, there've been severe bias where, for example, we talk about a physician who will prefer to have certain patient to be put into certain group of treatment rather than somebody else. But because of this severe bias, and sometimes patients were even bribing their way into being placed into the treatment groups. But randomization gave the clinical practitioners, clinical researchers an opportunity to understand that this is actually a process where there is no bias. There has to be an equal chance for every participant to either be randomized into the control arm or to the intervention group. So think about randomization like a coin toss, right? Where you can have a coin, you toss the coin, then you flip the coin, whatever side comes up, that's going to be your chance for participation. So the randomization is the best method for achieving comparability and is the basis for statistical inferences. So because every clinical study have to be, as we're collecting data, we make sure that data is actually have statistical analysis. And for you to conduct statistical analysis for your study, you want to be able to make sure that the study is fair. And how do you make sure a study is fair? You want to make sure that they're providing that accurate randomization ability to the study that will also enable the FDA to evaluate that the study was conducted fairly without any basis for bias. Now, why do we conduct randomization? If a difference in outcome occurs, so at the end of the study, there are two different, say you're looking at the intervention group and a control group, you're not able to know, okay, what is the possibility of that control group? Does it mean that the control group had more outcomes, more severe outcomes, or did we actually meet the objectives of the study? Because every study has an objective, and they are looking at the hypothesis for the conduct of the study. So in order for the study to be concluded, or to be effectively concluded, for a study that has two arms, they were thinking about having randomization. Now, it doesn't mean that every study that has two arms will require randomization. There are some studies that have two arms, but now the sponsor may choose to enroll a particular group of patients in one arm and a particular group of patients in another arm. Now, that is not biased. The way that actually works is the study design. The study design could be to be designed either a parallel. Remember, we went through the study designs last lectures. The study design could be parallel or could be split. So if the study design is split, then we're looking at the randomization. We're looking at where the ability where the study, for example, every patient will get enrolled. So once they get enrolled, now they split. They get enrolled, and now they split into different groups, into group 1 and group 2. And the reason most, of course, is that they want to be able to give, the study want to be able to evaluate how the interventional drug is related to the subject's disease or condition, and also how that can actually meet, how that can actually now compare with the subjects that do not take those interventional therapy, which of course will be the control arm. Now, just understanding what randomization is, we're just looking at the diagram for a study that comes in. Now, when a study is being conducted, we're having a screening, randomization, treatment, and outcome. So after the screening phase, for example, they're going to do blood work, or review physical exams and other x-ray scans, whatever the protocol requires. Now, after screening, which means that the screening phase is gone through, everyone goes through that screening phase. The next thing now is the randomization. The randomization phase is where they actually select to choose between where are the candidate participants going to. Because not every patient that is screened actually gets randomized, right? Because when a patient is screened, they can either be considered screen failure or screen pass. Screen failure is a subject that gets screened, but because of one thing or another, we realize that the subject is not eligible for the study. For example, part of the screening process will require collection of blood samples, physical examination, maybe EKG or scans, anything that the protocol requires. Now, when the blood results come back, we notice that instead of the blood, because the protocol will specify the actual measurement that they require for each test. For example, the protocol may require that hemoglobin A1C should be a minimum of 7.0, but a patient comes in and has a maximum of 7.0 for a diabetes study, which means that they want a patient, a minimum of 7.0, which means that they want patients that have high hemoglobin A1C, right? Hemoglobin A1C indicates your level of sugar, of course, which is your diabetes. Now, if we have, means that if study wants patients that are coming in with 10, 11, 12, 13, 14, which is really, really high sugar. But now, because we are doing this screening process, we screen a subject A came back and the result is 6.7. A1C is 6.7. Is subject A eligible to be in the study?
No.
No, they are not eligible to be in the study, so subject A will be considered as a screen failure. Subject A will not be randomized. But think about subject B, whose A1C came back as 9. So subject B is eligible because it's above 7. So now subject B will go through the randomization process. Now the randomization process now will mean that the sponsor want to find out, okay, now we've had this subject who is eligible. How are we going to look at this subject? Do we want the subject to be in the control arm, or the subject is going to be in the treatment arm? In order for us to give everybody a fair chance, let's find out what is required. Because the sponsor cannot just go in and say, oh, you're gonna go through I'm1, you go through I'm2, because the FDA will ask you, how do you justify who goes to I'm1 and I'm2? You have to give people a fair chance. Now, because the sponsor will not have any form of justification to determine who goes through I'm1 and I'm2, now the question goes to, now they have to create a fair opportunity where everybody will have the same expected outcome. Once this patient has been randomized, now they move to the treatment phase. Any study that has randomization will definitely have treatment, will definitely have more than one treatment, right? They will have a treatment and they will have a placebo. They may have a placebo. There are times when randomization doesn't necessarily mean that the study will have placebo. There might be more than one treatment option. There might be treatment A, treatment B, treatment C. right? Those are all active ingredients. And in their study, the sponsor may desire to want to learn which of these active ingredients is more effective, amongst patients. So they may look at that study population and say, okay, drug A, drug B, and drug C, we are testing all of you guys together. But we want to be able to see who actually gets more impact by expected outcome. So now when they go through the treatment, they will go through outcome assessment. Of course, outcome assessment now is the ability for them to actually, is ability for the subjects to be, actually, once the treatment is complete, then they're able to assess and evaluate how the patients are doing. Does that make sense? Or any questions? Okay. So basic clinical trial design. The sample, we have a sample here, we have randomization, then we have 2 expected outcomes. So if we're having a randomization and blinding, then we're making sure that we're having 2 outcomes, because we want to be able to see the outcome for when subjects actually get in the treatment and the outcome for when subjects get a placebo or any other form of therapy. Randomization and bias. So most common randomization methods include fixed allocation, simple randomization, block randomization, stratified. So these are all the different randomization process types that are involved. Each study may choose one form of randomization to go to, or some may even choose more than one, just depending on the complexity of their study and which of the analysis and how they are looking at. So 2 forms of experimental bias that are of main concern. So selection bias, of course, when the allocation process is predictable. So of course, allocation process is predictable, you're selecting the subjects. Accidental bias arises, even though you may consider that to be accidental bias when investigators say, oh, I didn't know that, I wasn't sure where to put the subject into. Then you end up putting them in the treatment, and because you consciously did that, or they may come back and say, oh, we didn't know anything. Then we're looking at fixed allocation. So fixed allocation, of course, assigns the intervention to a pre-specified probability. So now, because of course they are looking at probability and the ratio, that's where you're able to determine, randomization. So generally, allocation of the intervention to the control group should be equal, of course. However, unequal allocation does occur. Where of any, they say for every rule, then there's an exception. So there's usually those very, very minimal events where misallocation or unequal allocation will occur. The various method of fixed allocation is achieved as simple, block, or stratified. So just depending on the different kinds of studies that the sponsor want to analyze. So in your role as a CRA, you will not have to perform, right? You will not have to review a certain group of all of these randomizations. What you be actively involved will be just you understand what randomization is, but also when you have a study, when a study is assigned to you, you have to know the kind of randomization that is being conducted. And the randomization type will definitely be, typically be in the protocol. You understand what typically happens is that your involvement during randomization process, right? The subject goes to the site, gets screened, then they randomize them. Now, in most cases, or in some cases, when a subject is randomized, what happens? They will sit the site will take the subject's screening data, put in the computer, click a button. The computer will either do some configuration, then send a particular number to the site, or once they send that number, that information goes to the sponsor, which may come to you, right? You as a sponsor, then you would then conduct that randomization at the back end. as a CRA, as a sponsor, you conduct that randomization at the sponsor site, then send a response to the site. So now you're only looking at you, and of course, there's always a sequence of they have that sequence, that metric that you follow on how to randomize subjects. So it's not like something that you just sit on your own and say, oh, how do I actually randomize the subject? So you just go in and just click a button. It's the computer generates a particular number or then that number will consider the subject ID. Because that is subject identification. That subject identification now will be what the subject will be using throughout their participation in study, of course, which may be either being that they are in the control arm or they're in the intervention arm. So some of the disadvantages, advantage of that is simple randomization is easy, but typically imbalance. So this is actually, if you look at the screen, we are looking at the body. Let's look at an example, the body mass index, randomized assignment of male and female, simple randomization of flipping a coin. Now, there might be, if you're flipping a coin, there are only two sides of that coin, right? Either they have the head or they have the head or the tail. And if you flip that coin many times, you notice that there's going to be a sequence of when you flip it like this, maybe you're going to notice that if you flip it like this, the head comes up. And these are also chances where there could be potential bias, because I had one study where there were just two groups, A and B. So it made it so simple that when patient A came, patient one went to group A. And patient 2 came, patient 2 went to group B. So you as the investigator, what will be in your mind when you're looking at patient 3? You are muted.
A. He's going to A.
Exactly. So in your mind, I already understand that, oh, because this patient went here, this one went here. There's a chance that the next person will go here, next person will go here. So now that's also another form of that's another disadvantage of using the very simple randomization process. Now, simple randomization process usually happens. Sponsors don't usually use this method because it's very simple, and it doesn't enforce any form of statistical complexity. So what they typically will use is something totally either the block randomization, which we're going to get into, but the simple randomization usually being performed at the site level. Say a site is conducting an investigator-initiated study. That is their study, where they are funding the study. Now, when they are funding the study, it's it happens there. It happens with them, so they can create something that very simple randomization method. So we talked about the advantages and disadvantages of using the simple randomization process. So next one, I'm going to be looking at the blocked randomization. With the blocked randomization, this is pretty much what the sponsors will use. The simple randomization will be mainly used at the level of the site during their site initiation, their investigator-initiated studies. These are studies that are being conducted just at the level of the site. The site may be the one to conduct those studies, but for sponsored studies, it's usually the blocked randomization process. So the blocked or permitted randomization process this method is used to avoid serious imbalance in the number of participants assigned to each group, and will be equal participants in each group. So this is actually a good, a better chance for them to be able to have this, because, for example, you're looking at a subject, a study may have, let me just go at the demonstration, we'll have it better. So a study may want to have 4 different groups, for example, with a block size of 4 for 2 groups. So they're looking at, if they want to have a block size of 4, which means that they're going to have, at least that the ideal will be 4. Now there are 6 possibilities of having those 2 groups meet 4 times, right? So the two groups, for example, A and B, now you want these two groups to meet 4 times, which means that they want to be able to have those 4 blocks, which means the 4 blocks could be identification for subject ID, right? Because subject identification may include a series of numbers. So you see that the block identification is having A B B B, right? They have A B A B. You have A B B A. B A A B. So these are just the different formats, the different possibilities that they're able to meet. When you have 1 and 2, right? If you have 1 and 2, there are only so many times that 1 and 2 will meet, right? Or for this example, if they had included C, A, B, C in this example, if we had included A, B, and C in this example, you notice that there would have been more than 6 possibilities, right? That would be more than, there would probably be 9 or 12 possibilities, because that will be A, B, C, C, A, B, B, C, so all those possibilities will come into play. So they use a 6 different formats to look at the 2 A's and the B. So this is how the sponsor thinks when they're looking at the different metrics for randomization. They are looking at what A and B will meet, how often they will meet, and if often they meet, so what will be the combination? The combination for A and B. And all of this process is something that they will design. Usually the IT team for that company will design this, right? Once the IT team design this, I will talk about the different methods in which they'll be able to, stratify that process. Now they'll put that process into the computer. They configure into the computer, right? After the configuration, it goes through those different metrics when they're actually assigning subjects. So when we're at the side level, you're actually making that selection at the side level on the computer. What happens is that it goes through that series of sequence before actually selecting a particular number. It may just run for like 10, 20 seconds. Then select one number and say, okay, this is the site number. And you notice that it's a very precise process. So there's very little room for errors. So what are some of the advantages of using the blocked process? You're looking at a blocked process, looking at the balance between the number of participants in each group. Of course, you know that each process has each group has a particular number of participants. Therefore, of course, the type of patients recruited for the study, changes during the entry period. Blocking will produce more compatible results, because of course, you don't want to have different subjects at different times enrolling into the study, but this actually provides an opportunity where the site will be able, the sponsor will be able to evaluate the sites and the number of patients and the type of patients that are being enrolled into the study. The disadvantage of this is that if the investigators become aware of the block size, some assignments are known within with some level of certainty at some point. So looking at the stratified randomization process, stratified is divided into assignment groups by some clinical parameters. So the stratified, looking at subjects that are actually within a particular disease center, where they could be divided into different groups, ages, disease severity as well. Stratified randomization requires that the prognostic factors are measured before or either at the time of the randomization. So what this actually means is that during the time of screening, during the screening time, each candidate or each subject is randomized, is screened, right? So when the subject is screened, according to the stratified randomization process, that they will use that patient screened data to select patients that are of particular disease group. For example, you may have a study that says, okay, we are only taking patients, we want to enroll all patients with pancreatic cancer, right? But now, for us to be able to section and properly treat the patients, Patients that are screened that have stage 4 pancreatic will be put into this group. Patients that have stage 2 we put into this group. Patients that have stage 1 we put into this group. So now we are dividing them according to the disease severity. According to the severity of their diseases, that's how we're able to put them into different groups. And that's also another form of randomization. So, of course, stratify assures equal number of subjects among the subgroups. Only 2 groups or, 2 or 3 groups are usually being stratified. But the most important thing is that you want to, the more groups you include, the more groups you include in the stratified randomization process, the more complex the study becomes. And this actually applies to all randomization processes, because the more randomized options, the more randomized possibilities that you provide, then the more options it's going to be for the for the subject to be randomized into. Of course, the more complex the study will also be. Looking at the example that we have here, in a study where one can expect treatment effect to differ with age, size, and sex, it may be possible to for us to have 4 stratas: male over 60, over 65, so male under 65, female, and male. So you see that there's all these possibilities, because as soon as you're able to have gender is also another factor. If you're looking at, okay, I want to be able to stratify my group within, I want to stratify my group between the age and the sex. Now we have male and female. Now, what's the ages? You're looking at above 65 or under 18 or under 65. So most studies are typically want to have people that will be above 18, right? But now you're looking at above 18. Now, for them to be able to properly randomize subjects, they will prefer or properly design the study. They may choose to have subjects that will be able to say between 18 to 50, or between 18 to 45. They're going to be under this group. Between 45 to 65, they're going to be under this group. So this is also another example. Suppose an investigator wants to stratify age and sex and drinking history. So between the different ages, he may have to choose between the different ages. Okay, from 30 to 39, male. These are the ages. Are they a current drinker? Drinking history. So now with this one, they're also going to have a little bit of complexity here, because now what happens is that not just male, they're also going to evaluate female who are 30 to 39 who are currently drinking, then male of 30 to 39 who are ex-drinker or who never drank. So they're also going to evaluate all of these strata. So the design has 3 by 2. So this is 3 by 2 by 3, right? So it has 3, which is the age. This is the age range. So we have the age is 3 metrics, sex is only 2 metrics, and drinking habit is only 3. Is 3. So if you look at that, we multiply that, means that there are 18 possibilities that can come up from this. Right, there are 18 possibilities that can come up from this. So these are an example. Just like this is an example of what I was just talking about: 30 to 39 male current drinker, 30 to 39 male ex-drinker, 30 to 39 male never drank. So then you have the same thing apply for female. Then you go all the way to the other, to other subject, and all the way to 18. The advantages of using this, of course, is if a center should have to leave the study, the balance of prognosis study will not affect the other studies. Of course, this actually because sites do leave studies all the time, where the investigator or the sponsor may either fire the site for misconduct, or the site may not be actually enrolling subjects, so they may choose to close the site. And if that closes, because every site that the study opens, every site that the sponsor opens the study in, they are actually having their statistical analysis, the evaluation, and expectation that the site will be able to enroll a certain number of patients that will contribute to an overall number of patients that they want. For example, they may want to enroll 10 patients in the study, or let's say 100 patients in the study, and they want to enroll that 100% within 10 sites. So each site must enroll 10 patients. Now, site A finds out that they are unable to enroll 10 patients. They can only enroll 2 patients, right? that will definitely impact the statistical analysis of the expected study, of the study's expected outcome. Because they are, according to the analysis and projection, they've actually attributed that they want to be able to have 100 subjects, but because of site A inability to actually enroll 100 subjects or 10 subjects at that site, that they can only enroll 2, it makes it difficult for the for the sponsor to evaluate the effectiveness of the study using their statistical analysis. So now, of course, which means that this process will also be impacted. We're looking at adaptive randomization process. Adaptive randomization process involves adaptive procedures that change the allocation possibilities as the study progresses. In contrast, simple and block methods are defined an allocation sequence from the start of the trial. So for the simple trial, from the simple and block randomization process, we notice that when the study starts, with that method is going to go through to the entire study. But with the adaptive process, just to explain that as the study goes along, things change, subject changes. So a subject may leave the study, subject may die, sites may leave, sites may not enroll patients. But how do you meet up? How do you make sure that you meet with that changes? Because those are changes that you probably expected them, but you probably didn't plan for them, right? A subject was going to leave the study. Maybe you targeted, you wanted to enroll 100 patients. You end up enrolling 100 patients, right? Now, because you enroll 100 patients, the study, you're excited, you close enrollment. Now, as you move to the next phase of the study, during treatment phase, 10 subjects just die or just lost to follow up. You're calling them, site is calling them, no response, no feedback, nothing, right? At that point in time, that has the subject's departure or the subject's exit of those studies from the study actually impacted the way that the subject actually gets treated, and they of course impacted the statistical analysis of the study. Baseline adaptive randomization procedures. So the probability of the next treatment assignment is altered on the basis of the previous assignment. So it's a bias coin design. Of course, the essence of the coin is of the bias coin design is a procedure where the probability of the treatment groups varies according to the assignment of the various imbalance between the groups. So this is just to say that for each randomization process that occurs, for each adaptive randomization process that occurs. So as the study moves along from one phase to another, they're supposed to meet. They're supposed to actually change according to that study, according to the way that the study moves. But now they are looking at also the probability that this can be a biased process, where if 10 subjects exit the study, if 10 subjects die, or if 10 subjects decline participation, or if one site just stop randomization, or you just decide to go off the study, what will be the procedures in place in order to make sure that there are no imbalance? So bias coin is a procedure discussed below a reference. So we have this, you can click on this tool on the link. Once I share this, you can click on this tool and take you to this link where you can have more information about having the t-test and the p-values. But these are all statistical values that once you are looking at this, these are what the sponsors are using to determine their statistical analysis that will be used for the conduct of the study. Advantages and disadvantages. So the baseline adaptive randomization process, of course, has advantages where the study protects against severe baseline imbalance, because there might be times where they're enrolling patients during the baseline. Baseline is the same thing as screening. So during the baseline or the screening process, there could be different probability where they having particular number of subjects come on the site, enroll into the site. Some get screen failures and some get moved into different disease groups, maybe because of their disease severity or their ages. But at the end of the day, it protects against that severe imbalance that may occur, because since it's an adaptive process, it will easily adapt to any sequence that the study turns into. Operationally difficult to carry out. Yes, it is difficult to carry out because it requires a lot of attention. Doing the adaptive process requires a lot of attention to meet those procedures in place for the effective study of the effective conduct of the study. Now we have the response randomization process. The response randomization process, of course, uses information on participants' response to the intervention throughout the course of the study. So think about a study where during the time of the study, they are doing a survey. The survey must be completed during the time of the study. And during screening, they will have to, they'll give this survey where patients will go in a check mark. How do you feel today? Check from a scale of 1 to 10. What pain level are you? 1 to 10, subject will click 5. Then they said, how do you feel? Sad, happy, moody, subject cycles. So now, once the sponsor collects that data, now they are able to use that data, which is of course considered response adaptive randomization process. Once they use that response adaptive randomization process, they're able to use that information now to be able to properly randomize the subjects. Play the winner. The procedure assigns the first participants in the toss a coin. So this is an example of this process where group A subject, of course, we're looking at 7 subjects here. Group A subjects in group A will be 1, maybe they've responded the way that they responded to their questionnaire, they assign them 1, and 2, this one assign them S and F, sorry. They may have success, screen success, and screen failure, right? If we have screen success for patient 1, patient 2 was a screen failure, then we're looking at, okay, patient 3 got assigned to group B. Then group B is also success for number 3 and 4, then 5, of course, a failure, then 6 and 7, respectively. I know that this section of randomization and blinding process could be a little bit complex. And the reason for that is because they are using a lot of statistical terminologies, which of course, you just have to familiarize yourself with. This is not something that you're going to be doing on the job, but it's a good thing to know what the process are and what the advantages and disadvantages are, because as you go into your job as a clinical research associate, there might be times where you have an opportunity of actually, your team lead may require suggestions of something. Maybe they're designing a new study and they need your input, or whatever, but at least you have that sort of background information, background knowledge of what the process is and the expected outcomes. 2-armed bandit. So a 2-armed bandit continually updates the probability of success as soon as the outcome of each participant is known. So this is actually a situation where not every subject that got into a study finishes at the same time, right? For example, we want to be able to enroll subjects for 12 months between January to December. We enroll patient 1 in January, on January 1st, patient 2. And the study has 10 cycles. Each 10 cycles will occur between the first treatment for 10 months. If subject A starts in January, there's a likelihood that they're going to end somewhere in October, 10 months. If subject B starts in February, they're going to end in November, right? And if subject C starts in March, they're going to finish the treatment in December. So because we enroll patients at different times of the study, it makes it difficult for patients to all complete their study at the same time. But of course, that's actually a known possibility, because you can never have all the patients complete treatment at the same time, unless they were all enrolled at one time, which of course is also difficult. So now, the way that the 2-armed bandit method actually works is that once we have a subject that completes the first study, for example, we have that subject that enrolled in January and finished in October, that subject now will be considered as finished. So now they'll go in there and actually perform the randomization process now for the study, for the subject, even though now, because they've actually projected initially at the start of the study, they've already projected that subjects will be enrolled at this time and they'll they'll be completing the study at this time. That's the randomization process that they're going to be using for that study. So when that patient actually completes the study, they're looking at the next patient. Means that it's easy to predict. It's easy to predict that, okay, in next month or next 2 weeks, we're going to have a certain number of patients that will be completing the study as well. This method was developed in response to ethical concerns shown by some clinical investigators, of course, because, of course, bias on the basis of bias. The disadvantage of this, an imbalance in the group causes some loss of power for the study. That's true, because now, if a subject completes the study, terminates the study prematurely, because the goal is that we're anticipating that every subject will complete the study. They will complete the 10 months treatment or everything that's supposed to be done. But if, unfortunately, for whatever reason, the subject decide to leave the study prematurely, then of course that just messes the whole thing for the study. Inappropriate randomization methods. So assigning patients alternately to treatment groups is not a randomization assignment. So there was a study that I had. The investigator initiated study. They were very lazy to initiate or to start sort of a randomization process. So the way the investigator said, he said, well, if we assign patient 1 on group A, we should just assign patient 2 on group B, and we should just follow that same sequence. And he says that that's the randomization process that will be that will be using. Easy, trying to make an easy fix. Well, assignment now, so they said that these are the assigning patients to alternate random treatment arms is not actually randomization. Assigning the first half of population to one group is not also randomization. So assignment by methods based on patients' characteristics is also not a randomization process, because you cannot base patients by their date of birth or by the clinic data they came into the clinic, because anybody can come to the clinic at any point in time. So mechanism for randomization: if the chosen randomization process is not implemented properly, then the entire randomization process can be compromised, avoiding advantages of using it in the first place. So of course, if randomization is usually not only for a multi-center trial, but also it could be a multi-group study, a trial that has multiple groups. So it's recommended that, the implementation, we want to make sure that we don't have any sort of randomization process where if it becomes very, very complex, right, it makes it difficult for the team to follow. Now, if the study because as soon as the randomization process is jeopardized, that's why it's very important for sponsors to take very critical look at what randomization process they're choosing for the study. Because as soon as they choose something that is not congruent with what they're looking for, with their statistical expectations, then it just misses the entire randomization process. And if that happens, they may have to put the study on hold until they figured out which method will be used properly for the effective conduct of the study. Now, what are some of the requirements in order for you to have a randomization schedule? So the randomization schedule should be unpredictable, of course. It means that they should not be able to predict that patients will be able to go into either group, because if it's predictable, then it doesn't make sense, because the investigators will be able to know that, okay, if I put these patients on group A today, I can definitely put them in group B tomorrow. So it will just be simple, the same way that the investigators have been doing it at their own site level. Appropriate balance, of course, there has to be a good ratio between either 1-to-1 ratio or a 2-by-2 ratio, because if you have a 2-by-1 ratio, what happens is that there's a possibility that you might end up having an imbalanced data. Extendability could be extended. Of course, randomization should be well documented, and of course, reproducible, which of course you have to duplicate the randomization process for other studies. Requirements for sound allocation sound treatment allocation schedule. Assignments remain masked to the patient, so just making sure the assignment of randomization actually become masked, which means becomes blinded to the patient, the physician, and any other clinical personnel that may be at the initiation of treatment, which means that, of course, they want to blind the treatment for each, patient. How will you think about a patient that actually comes in and say, I think I know that I'm getting the drug or the placebo. So sometimes patients can tell, but they are telling it's not, it's not actually a definitive. They can tell from just what they think, right? But it's not something that they can well say, yeah, I'm sure that I'm really just taking I'm sure that I'm only taking the pill, or I'm sure that they cannot be sure for anything. Also the clinic personnel, think about a person who works at the clinic as the site coordinator or the research nurse. They can only have that thoughts in their mind, but they cannot be sure that actually this is something that this is the kind of treatment or this is a treatment arm that the patient is being assigned to. Prep for randomization schedule. This is a typical situation. So schedules are prepared in advance and kept in safely. Of course, the computer files. All this, like I was mentioning, before any randomization process takes place, this is something that has been written down, documented, created, and I've seen all the mathematical jargons and possibilities that could happen in that study. So once they have all of those processes written, they have it audited. So once they have it audited, then they can actually have that process computerized, where they will be able to, so they start having the process computerized, where they'll be able to now start looking at what are the possibilities of each scenario. Allocation concealment. It is very important that those responsible for recruiting patients into a study are unaware of the group to which a participant will be allocated. That should agree, that the subject should agree in the study. So if you're having the subject to enroll into the study, it's very important that, the person who is randomizing the patient, think about Mr. Ndasi Elvis. He's working at the site level. He's a research nurse. The patient comes in, he screens that patient, and now he wants to randomize the patient. Do you think it would be fair for Mr. Ndasi to know that, oh, patient A will be going to treatment arm, or patient B will be going to non-treatment arm? So it is practically impossible for them to be able to do that, because if they do, then the whole randomization process has been jeopardized. So it's for a multi-center site study, central randomization by telephone or IVIRs. So they usually ask what kind of method, or a common question that you get during your site monitoring visit will be that, what method of randomization will be used during the study? So most method of randomization is the IVRS. IVRS, interactive voice response system, where you'll be able to just call a particular number, then you press the button. But recently, all randomization process that I've recently encountered has been over the internet. The last randomization process I did over the phone was in 2016. That was the last randomization process I did over the phone. Everything else now has been on the computer. Types of randomized studies. So we have parallel group, sequential group, group sequential trials. We have crossover, and we have factorial study design. So these are all study designs. Like I mentioned earlier, each study design will have a different particular randomization parameters that will go into them. So parallel group, you think about two groups randomized, treated and followed as two groups response. So measure at the end of the study, of course. Now they measure the data according to the percentage from baseline. So a common thing that I've noticed, even in my job, is that when they're asking one question, when you want to measure the patient's safety, are you measuring it from the time that the patient actually, from the previous visit, or you're measuring it from the baseline visit. So for example, you want to be able to measure a patient's hemoglobin A1C, right? Are you measuring it to assess the patient based on the fact that the patient had 7, A1C was 7 milligrams per deciliters at baseline, or for example, you're evaluating it from a previous treatment or previous study, previous treatment that the patient had. For example, they may have a visit that occurred yesterday and they have another visit today. Are you going to evaluate it from the visit that occurred yesterday, or you're going to evaluate from the visit that actually is going to be occurring today? Sequential trials, not for a fixed period, of course, terminates when one treatment lays up the superiority. So there's, sequential trials, of course, when it's not really when they don't usually allow the study to go through to the end. What usually happens is that when a study is being conducted and the sponsor sees, because of the data that they're collecting, they're able to evaluate to see, okay, we're having more patients are actually being randomized into this arm than this arm. If they expected that you're going to have 45% of patients enrolled in this arm and 55% of patients enrolled in arm B. So 45 on arm A, 55 on arm B. But eventually what they see is that when the study is being conducted, you notice about 70% has already been enrolled into arm A. So they just end up just assuming that, oh, arm A is the superior because we expected arm B to take that 55%. But if arm A has already taken 70%, there's a possibility that arm B will not be able to recover from that. Group sequential trials. So they are popular, they analyze data after a certain proportion of results are available. So of course, when a certain proportion of results becomes available, this is when, when we're reporting to the FDA, we are either asking the question of, okay, do we want to report to the FDA when the study is completed, or want to be able to report to the FDA when the study is not completed, right? So at different times of the study, at different times, we can either report this to the, report the study to the subject. You can report the subject to the study. Give me 1 second. So early stopping happens when, of course, one study, of course, has been declared, having clearly indicated as a superior. So just like what we had at the sequential trials, the group sequential trial, of course, means that they're having more groups of patients coming in. Crossover trials. So we have two groups randomized. Each starts with one treatment and then the designated time point, then cross over to another study. So typically what happens here is that we have studies that a study will begin. The sponsor may plan that the study will actually start and run for like 1 year. And when that study terminates, the patients will now move out. The patient will now move out now to the next study. So that's called a crossover design. Now, when they cross over to the next therapy, of course, the new study may be that they have a new drug that they're going to be starting. But now, what are some of the things that are involved? It means that they have 2 treatments. They have 2 treatment periods over, and of course, each patient has their own control of either moving to that study, into a new study or not. Now, there's a washout period. Washout period means that between the time that the patient started taking drug A to when they are moving to drug B, they have to be, for the study to be effective, for the sponsor to effectively analyze the data for that patient, they have to first of all go about and make sure that the patient eliminates, or you're doing that washout period. The washout period may be a period where the patient does not take any drug. The patient may be for like 1 month without any drug. so that the drug that they have taken for the previous study will just digest and clean up from their system, so that when they're starting the new study, they can actually better analyze the effect of that new drug. Now, for some therapeutic areas, think about an oncology therapeutic area, where they may have new studies, they may have a washout period where the patient may have infusion of saline water just infused, and they can go, that saline water now will go in and be conducted as a washout period where the subject will be washed out for that duration. Give me 1 second. It's an example of a washout period. So we have the sample. We have the sample here. The patient got randomized into intervention and placebo. Now they have the expected outcome here. Now the expected outcome, they're thinking of, okay, now they want to cross over the study. During the washout period now will be for the patients they were taking the intervention. They're having that period where that period may be 1 month, 2 months, or whichever duration according to how the sponsor design it. During this time, the patient is not taking anything. The patient is not taking any active ingredient. And also for the placebo patient, likewise, they are not taking any active ingredient. Now they're crossing the patient over. Now you that was in patient A, you that were taking the intervention, you will now move to start taking the placebo. And you that was taking the placebo, you now move to actually start taking the intervention drug. Now they've actually analyzed the first outcome here, right? Here they've analyzed the first outcome. You were first of all taking intervention, and you were first of all taking placebo. Now we've actually had this result. This actually run for like 6 months or 12 months. Now they've taken the first result. Next, they're moving on to the next phase. The next phase we're studying now that you that are taking the intervention, now you're taking placebo. Now also they're going to have the expected outcome here as well to look at the results. Once they look at the result, they're able to determine, so if you actually had this outcome, you actually had this result, so what's the possibility? Now they're able to evaluate their result to see which treatment was better and how the patient responded to each therapy. They may end up realizing that patients that were enrolled in group A intervention arm actually are better, because the patient doesn't know that they are enrolled in either arm or not. They just know that they are participating in the study. But this at the back end. At the back end now, the sponsor is the one evaluating and looking at this and say that, okay, this is how we're actually switching the subjects over from one arm to another. The factorial design becomes a little bit complex, because of course they may have treatment at different conditions. They may either have to use treatment and also the condition. So it stands for intervention and of course placebo. So we have looking at intervention A and B, we have the outcome. Intervention A and placebo B have the outcome. Placebo A, intervention B have the outcome. They have placebo A and placebo B have the expected outcome. So these are all the different factors that may be involved for the factorial design. Now for incomplete factorial designs, looking at a nutritional interventional trial, these are trials that may be used just for like nutritional purposes. These are typically trials that will be conducted for typically by NIH grant-funded trials, where they have their own metric of analysis, but it's definitely not something that sponsored pharmaceutical companies will be conducting. Bias. Bias may be defined as a systematic error or difference between the true value that actually obtained due to all causes other than sample variation. Some potential sources of bias, of course, patient bias, we have provider bias, assessor bias, laboratory bias. So bias could actually be conducted or experienced at every level of the conduct of clinical study. Patient bias. So the patient's knowledge of the treatment being received may affect the outcome of the study. So of course, we don't want the patient to know that they're taking the intervention drug or the placebo, because once the patient becomes aware, then the whole study has been jeopardized, because the patient may choose to leave the study or go about and start marketing or start talking about the study prematurely to their family, or which of course is not something that you want. Care provider bias. The care provider bias, the care provider's knowledge of which treatment a patient is receiving may also affect the treatment. Because think about a provider who may be able to see a patient and say, oh, he knows that patient A is going to receive an active ingredient, and patient B is not going to receive the active ingredient. He may choose to switch theirs, but once they become aware, and also he may, given that he knows that the patient will be receiving a particular group of patient will be assigned to a particular group of study drug, either the placebo or the intervention, he may choose to withhold enrollment for a particular group of patients, for a certain patient, because he knows that, oh, wait, when we have a next patient, then we're going to have you. When we have this patient enrolled, they will have you to enroll, because you know that when you enroll, then you're going to have the intervention therapy. Assessor bias. The assessor's knowledge of treatment, of course, of the treatment the patient is, of which treatment the patient is receiving, may affect the assessor's outcome of the study. Such bias will directly affect the validity of the conclusion of the study. So this actually assessor where you notice that at some point, this study is going to be unblinded, right? At some point, we want somebody to get to know which arm the patient received, or but we don't want them to know just yet, because everybody else that we're looking at, we look at the patient, we look at the provider, we're looking at assessor. Assessor might be somebody who does like the scans, right? Who does basic scans or CT scans or MRIs for the study. Laboratory bias. So this laboratory bias, of course, goes along for the subjects that have, just going through the lab as the blood results and samples have been collected. Now we're looking at, okay, if the samples have been collected and interpreted and tested, now if the laboratory technician knows that the patient is receiving either the randomization arm, either the intervention arm or the placebo arm, they're able to evaluate to know the kind of expected result that the patient should have. Now, analysis and interpretation. So, seeking an explanation of an anomalous finding should also be contrary to the study hypothesis. Now, the study is being conducted. After the study has been conducted, now we're looking at the analysis. Study has to be analyzed. The statistician may be the same person who did the randomization procedures, who documented all the randomization procedures. He or she may be the person that evaluates to see, okay, I know what's happening here for the study. I can evaluate the study from here. But now it might not be time for them to actually crack the code of randomization, right? He or she may not be the one responsible for cracking the code for randomization. They might, data has been collected and is sent over to this person for them to analyze. If they become aware that a particular group of patients were taking the placebo and a particular group were taking the intervention, they know which groups, then the level of their commitment, and the complexity of their job, will not actually be meaningful anymore, so to speak. Because, say, for example, terminating a study for adverse event because they fit the expectation of the monitors, terminating a study. So the knowledge of the treatment group may affect the decision made by external monitors of a particular study. So knowledge for a treatment group, because if a study now gets treated, if a patient is being treated, the sponsor who comes to the site, you as a monitor comes to the site periodically to evaluate the study, to go to the site to evaluate the data and the rest. Now, whatever you are seeing, you as a monitor, when you go to the site and you see that there are so many adverse events coming out from the patients from this particular group, what do we think? You may think that, okay, let me just go ahead and actually add them an adverse event. Because now, if you know that the patient is receiving the placebo and there's an adverse event, you may just feel like, I don't need to, maybe I don't need to report this because it's just an adverse event. And of course, the patient is not even receiving the active ingredients, so maybe nothing to worry about. But if you are not aware, you will consider every adverse event to be very, very serious, because you don't know which is actually being interpreted or being affected by the outcome of the study. Volunteer bias. It can be defined as a bias that occurs when a particular sample of the participants drop out or remain in the study depending on their willingness. Sick patient in the control arm may drop out of the control due to death or other illnesses, of course. Patients in the experimental group might lose their interest in the group because the improvement of the result does not affect. So volunteer, again, is patients that comes into the study. They are volunteer, their willingness to sign the consent form and enroll into the study as their particular personal willingness to do so. Now, when a patient become aware that they're actually receiving an intervention drug, right, and they are not getting well, then they feel like they're going to take off the study, they're taking off from the study. If a patient become aware that they're actually taking the placebo and they are not having any sort of response, they want to get off in the study. Surveys have volunteer bias due to willingness to part of their individual of an individual to answer the questionnaires. Of course, patients may desire, because the fact that patients come to this to the participate in the study, it's usually not the immediate benefit for them, right? Because on the consent form, the language in the consent form usually states that your consent to this trial, this drug will not impact you directly, but may be contributing to science which may impact future generations. That's usually the language that you see in most consent forms. And the reason they're having that is because just to emphasize that the patient will not be getting that, will not be getting that treatment, will not be getting that particular clinical benefits from that therapy, from participating in the study, but this might be something that they want to also look into. Design techniques to avoid bias. So what are some of the design techniques that are used to avoid bias? One is blinding. Blinding is intended to limit the occurrence of conscious or unconscious bias in the conduct and interpretation of clinical trial. Double-blind trial is the standard. So usually a study is double-blinded. A double-blind trial, the treatment should look, feel, and taste the same, and no one should know about the treatment assignments. If the sponsor staff is required to be unblinded to the treatment code, the sponsor should have appropriate SOPs to guide against the inappropriate designation of treatment codes. In some cases, open trial ethically and practically possible. Of course, there are times where they might be conducting studies that are open trial. Open trial means that, open label, the group is known, the disease group is known to the patient, the various drug groups are known by the patient, but not necessarily mean that all open trials are biased. But just like the word said, blinding, right? Blinding means actually being prevented, which means that the sponsor will the subject will not be aware. Sometimes the site is also not aware, which the investigator is not aware of which treatment that the patient is actually being treated upon. Breaking the blind. So breaking the blind, which means that if for any situation there's an emergency that needs to be conducted, then the patient and the investigator realizing that, oh, you know what, we need to break the blind for this study. I need to know what kind of patient, what kind of drug the patient is getting, so that I can properly treat them. For example, you're treating a patient, and the patient, patient experiences swellness in the neck, or abscess. Patient experience abscess, and abscess is not part of the expected adverse events in the investigation brochure. It becomes very severe. grade 4, patients about to die, you know, it's like this is the very patients hospitalized and this is severe. Now, what happens is that the investigator will reach out to the sponsor and say, hey guys, I need to know what kind of treatment the patient is receiving. Either they're getting a placebo or they're getting the active ingredient. If they're getting the active ingredient, I want to know if I'm going to stop them from this therapy. You know, I had one patient, I had a study with one physician who she was very cautious. She's very cautious about her patients. She cares about it. If any patient just exhibits diarrhea or anything abnormal, she just stopped the therapy. She stopped the therapy so often that the sponsor came and said, let's amend the protocol to say that if you stop the therapy due to an adverse event, you can actually restart treatment again after the adverse event has actually gone away. Because what they typically had to do, what they did in the past, or what they did initially, the way they designed the study initially was that once you get into the study and you stop treatment, then you can no longer restart treatment. But because of this, she was actually made it possible for the study to realize that they have to start restarting treatment for patients that may have stopped due to adverse events.
So I have a problem with these adverse events.
Come again?
So the adverse events, I think they can unblind the study. If another group will frequently see adverse events and on another group we don't see adverse events, that can easily unblind it, right?
So you said that unblinding is usually for a single subject, right? Unblinding doesn't necessarily mean that they're unblinding for the entire study. Unblinding will usually occur when the investigator realizes that, you know, they have a severe adverse event in a particular patient that's happening. If that happens for all the patients, then it becomes a much more bigger concern. But if it's just one patient going through this, then they have the ability to request for unblinding, so they can know what treatment the patient is receiving, so that they can better assess them clinically. So all of these potential problems can be avoided if people involved in the study are blinded to the actual treatment that the patient is receiving. Of course, blinding, also known as masking. Degrees of blinding, rigorous blinding procedures for randomized controlled trials are essential to minimize bias, just like we've been talking about. So just to understand blinding in a more distinct manner. So types of blinding. Open label. Open label means that there's no blinding. The patient is aware of the drug they are receiving. The subject is aware, the site is aware, everybody's aware of whatever each person is receiving. Single blind, the patient is blinded, usually occasionally by the assessor. So this usually doesn't occur, only single blind. There are single blinded studies where the sponsor, makes it that only the subject is not aware of which arm that they're receiving, but the investigator is aware. Double blind, patient and the investigator assessor, who often are the healthcare providers and the data collectors, are blinded to the treatment. It means that everybody is blinded. Complete blinding. So complete blinding means everyone in the study is blinded. Of course, when we talk about everyone, we mean the investigator, the subject, the data person, the CRA, everyone is blinded, right? But for a double blind, only the patient and the investigator are blinded. You as a CRA, you will not be blinded because you are working for the sponsor, so you know that the study is not blinded. So this is just full explanation again about the open blinded study. Open label study, of course, it's in unblinded or open label study, both the participant and investigator are aware of the investigation of the investigation that the participant has been assigned to. The single blinded studies are usually done with the blinded study, just the patient, the healthcare provider usually know the treatment that the patient is receiving. And the advantages of using the single blinded system is that it's simple to carry out. Of course, knowledge that the investigator may help, it helps the investigator to assess the patient better, to know what kind of method to be able to use. But of course, the disadvantage of this is that it also may have a potential of causing bias. because if the investigator is aware that the patient is receiving a particular drug, that's just the whole point. We don't want them to be aware. But of course, if this happens, then for a single blinded study, it means that there are different metrics that they're looking at, which the bias is always of paramount importance. But but this is something that they have to pay close attention to. A single blinded trial. So this is just an example of a single blinded trial where you can easily know. So single blinded studies, you guys can take a look at this and understand what's happening of the single blinded.
Hello, Andrew.
That's true.
I did not hear. Okay, I thought it's my system that cracked down.
Sorry, come again.
No, I did not know you step out.
Oh, sorry.
So no break today.
We are, what time is it? 9:13. I want us to go through this blinded. Okay, we have about,
Let's finish.
we have about 10 more slides.
Okay, let's go ahead.
Okay. So this is just an example of the single blinded studies. You guys can read on this example, just more on this. The single blinded study, we have the aim. You're looking at, most important thing that you're looking at a study, right? You're looking at the aim, the design. Study design is very, very important. The settings, then the methods, result, and conclusion. This is not how a typical study would look like, because usually very intense and usually very big in a protocol. But this is actually an example of a single blinded study you can just look into. For a double-blinded study, when both the subject and investigators are kept from knowing who is assigned to which treatment, the experiment is blinded, it's double-blinded. Of course, the investigator is not aware, the subject is not aware, so that becomes double-blinded. So double-blind matching of drugs. If the tablets or capsule are imperfectly matching, there could be a problem in blinding. So usually when a patient, patient can easily identify the drugs by either they know the shape and color of a particular drug. For example, the sponsor in the investigation brochure will usually have the design of that drug, which may have a particular color, red, pink, whatever. And the way the placebo of that drug will be, it may be similar or may have a different smell or odor, which may, of course, differ. You may easily identify which drug the patient is receiving at the level of the site. Special problems with double blinding is that maintenance of blinding makes the study expensive, because double blinding becomes very expensive for the sponsor. The sponsor realizes that, okay, we are having, we are conducting a study, and it's double blinded, but they becomes expensive because they need to assign more people into the study and more people into maintaining the integrity of the data that has been collected at the level of the site. And also when a site monitor, a CRA like yourself, go to the site to review the data, which is called source data verification, when they go to SDV the data, it becomes increasingly challenging for them, because They have to pay closer attention to making sure that the study was conducted properly, the site is actually matching the protocol. There was no unethical conduct of the study performed during the whole time that maybe a patient has been participating. And psychologically speaking as well, there might be times where they have different scenarios that are occurring for the study. But for a patient that's actually receiving a particular drug, or for that double blinding, it makes it difficult for the sponsor to manage, because now they need more people to coordinate the efforts. Stumbling block, side effects. So side effects, of course, observable by the patient are much harder to blind. They're harder to blind because side effects, of course, this is events that happen as the subject continue to participate in the study, right? Efficacy, a truly effective treatment can be recognized by its efficacy in patients. Now, although rare or some new treatments truly are major leaps, when this happens, it's usually very clear that the treatment a patient is receiving, at least. So when they're looking at the result, the expected outcome, when the outcome occurs, when they actually have the treatment result able to understand the efficacy, which is the effectiveness of each drug at the during the study. Now, when the result actually come out, now the investigator will be able to know that, okay, this patient was actually receiving this drug. But at least it's better for them to receive it at the end of the study than to receive it during the study. So I'll let you guys, the example of a double-blind study. You can go to the NIH website to read the example of the double-blind study and how that actually entails. Triple-blind. Triple-blind is an extension of a double-blind study. The committee monitoring the response of variables are not told the identity of the group. So there's usually something called DSMB. DSMB is Data Safety Monitoring Board. Yes, Data Safety Monitoring Board. So a DSMB is a particular group of people whom they may just sit, a particular group of people. These are physicians who are seated somewhere, and they are reviewing safety concerns and the response of the subject to this data, to the group, to the drug. Now, once they are able to look at that response, now as they're looking at that response, maybe an opportunity for them to say that, okay, let's look at the response, let's see how the patients are responding to the study, and they can actually evaluate that. But for a triple-blinded study means that the DSMB guys will also not have an opportunity of reviewing that. They will also not be able to know which patient has been assigned to each drug. So I'll give you guys also another assignment to review this on the NIH website, and this is an example of a triple-blinded study. So once you review that, then we can discuss for any questions that you may have for the randomization process. So you have the first assignment to look at the double-blinded website and also on the triple-blinded website. Unblinding the trials. So unblinding the trials, the drugs can occur either due to an error by the personnel of the trial, or might be an official blinding. So if it's an error, of course, that would have some repercussions, right? But of course, in an emergency situation where the investigator, just like we've talked about, the investigator notices that a patient is having an emergency, and they request to know the particular drug that was used for the conduct of that study, then they can actually request the sponsor to provide the code to unblind the study. So once the sponsor provides a code to unblind the study, then they become aware that yes, actually once the site become aware of the drug that the subject is receiving, then they can properly decide how to treat them clinically. So the major takeaway here is that The manner in which the chosen randomization methods is usually implemented are extremely important. And the randomization process, the randomization choice that goes into the study matches along with the study design. So for example, if they have a parallel study design, they're going to have a parallel randomization process. Now, think about the unblinded, blinded, single blinded, double blinded studies. So for all the different blinding procedures for the studies, it means that the sponsor will have to choose which of the, and first of all, before they choose which randomization process to select, they have to look at their study design. They have to look at the objective of the study. Once they look at the objective of the study and the study design, then you see what randomization process will be the best for them to be able to practice, to eliminate bias, meet the patient's expectation, and also meeting the objective of the study. So this brings us to the last slide of this lecture of randomization and blinding. Questions, please.
So my question goes on triple blinded study. So who's the third party who's blinded?
So we're talking about the DSMB, the Data Review Committee Board. So the Data Safety Monitoring Board. So the Data Safety Monitoring Board are the group of guys who they are physicians, male, female, they are physicians who they review the safety of the study. It means that when any adverse event occur, a death occur, a major event occur, they send it to these guys, right? These guys to review it, they assess it, they evaluate to make sure that this study is actually the adverse event or the serious adverse events was part of that study, or or was attributed to that drug. But now, if they are not aware, it means that they are also blinded, which means that we have the subject is blinded, the site is blinded, and the DSMB guys are blinded.
Okay. Because if they are not blinded, they can easily draw conclusions that will bring a big bias.
Correct.
So
But for most cases, though, the DSMB do not actually participate. The DSMB are actually working for the sponsor.
Okay.
So they are not at this level of the site. They are the sponsors group. So the sponsors group now will become aware of the drug and become aware of what's happening at the level of the site. Yeah.
Okay.
After unblinding, to solve the situation that occurred, can the blinding take place again?
No. So when a blinding occurs, when an unblinding procedure occurs, when that adverse event emergency happens, where the subject the sponsor said, the doctor say, you know what, sponsor A, I need to know the drug that you actually getting during this patient, if they are receiving the placebo or the intervention, or it's an emergency situation. Now, once they unblind that drug, for the patient to get treated, they can no longer blind it, because once that patient gets treated, it's likely that the patient will no longer be in the study.
Okay, is that because you said it is a case-by-case unblinding? They don't do general unblinding?
Correct. It's a case-by-case unblinding, and it has to be very emergency situation. Because now, once they have that, once that emergency situation happens, then they know that, okay, this is actually major, and the investigator, they reach out to the sponsor. The sponsor also look at their own analysis and look at their own criteria for unblinding. And once they have their criteria for unblinding, they can go ahead and say, okay, yes, go unblind the study. Once they unblind the study, then what happens now is that the investigator and the site and investigator now becomes of the procedures for that particular subject.
You said they can recruit 110 persons die or withdraw, and they're making communication and no way to get them. What percentage of such drop can stop the process?
It depends on the sponsor and the protocol, because there are patients that will be lost to follow up. every study have expected result and expected outcome, and it means that for a study to be conducted, there are times where they may say that, okay, we know that even though we desire to enroll 100 patients or 1,000 patients, there's a possibility that 10 patients will be lost to follow-up, or 20%. They have that percentage that they're analyzing to know that, okay, this is actually a particular percentage of patients that we know that they will be lost, and they can prepare for that. But if the percentage exceeds, then it actually disrupts their whole statistical plan. But in order for them to be safe, they can just increase the percentage of expected screen failures or patient exit, or all those things. Then if they increase that percentage, then it's just like hoping to fail, but hoping to pass but expecting to fail.
The right word to use if asked this question anyway is it depends on the protocol of the project.
Yes.
Okay.
And the sponsor too.
Okay. When we when we say and the sponsor, is the sponsor not part of the protocol process? Is this protocol done out of the consent of the sponsor? I think the protocol is that document that binds the sponsor and the whole thing down, that is laid down before the investigation begins.
Correct. The sponsor is so a couple of things, right? The sponsor can be a person that funds the project, right? Might not be the same pharmaceutical company.
Okay.
And the pharmaceutical company, the sponsor may say that we don't want blinding. They are bringing the money, right? The pharmaceutical company or the protocol writers. Okay, so for example, I have my job now. What's happening is that we are doing something called investigators contribution projects, right? Now, what happened is that we've assigned different doctors of different capacities to write protocols. They write the protocols, they bring that protocol to us. We are the sponsors. We have the funding, we have the money to fund that research. But now, once they conduct the study, once they write the protocol, they bring the protocol to us. We are reviewing the protocol. We review the protocol. If we say yes, let's go ahead. They go ahead and start conducting that study using that protocol. Now, we, the sponsors, may decide to say, okay, do we want X, Y, and Z? The protocol writer is that doctor. The doctor may say, yes, according to according to this protocol, I was able to have this protocol based on X, Y, and Z values, right? I'm putting A, B, and C on this protocol because I am evaluating patient, I'm evaluating insurance, I'm evaluating this and this and this. They may have their reasons for putting all of everything, but they have to be that justifiable reason, justifiable discussion between the sponsor, the protocol agreement, and the site. So not all cases where the sponsor is the protocol developer. There are some cases where the sponsor will be the one that produces the protocol. They have the protocol team in-house, they do everything and produce and send to the site. But there are cases where the sponsor do not have the capacity to either write that protocol, to develop that protocol, but they want to develop their product line. What they end up doing is that they can outsource the development of that protocol. And now the investigators who are involved in writing that protocol or developing that protocol, they will be considered as national PIs. National PIs means that there will be people that all other doctors can reach out to for the conduct of that study. They'll be like the lead. You are muted, Pastor. I cannot hear you.
I said, then that's right. I understand now. I'm muted because I do not want background noise to get into my class.
Okay.
Yeah, it's understood. Thank you.
Okay. All right. So thanks, guys. I think we can take a break. I'm gonna end the recording.